We have everything.
Okay, we'll let people in.
You should mute your computer just in case.
Okay, we have Facebook live streaming. Letting people in. Why can't we see these? All right, can everyone hear me? Great. Hello, everyone, and welcome today to our seminar. I will start by reading the land acknowledgement. We respectfully acknowledge the territory in which we gather as the ancestral homelands of the Beothuk and the island of Newfoundland as the ancestral homelands of the Mi'kmaq and Beothuk. We would also like to recognize the Inuit of Nunasiavut and Nunatuhavut and Innu of Nitsansian and their ancestors as the original people of Labrador. We strive for respectful relationships with all the peoples of this province as we search for collective healing and true reconciliation and honor this beautiful land together. It is my great honor to introduce Dr. Julie Schramm, who will be talking to us about food web dynamics and from Antarctica. So Julie, whenever you're ready, you can go ahead and share your screen.
All right, can you see the Nudibranch?
I can.
Awesome, okay. Pull this up. All right, so thank you very much for inviting me to discuss some of my research today. I will be focusing on some of the research that was part of my PhD work and then follow up with some of the more recent work that I have done to investigate responses to climate change. So to get started, I just want to give you a brief outline of what I'm going to be talking about. First, I'll start off with an introduction to the study area and some of the study organisms. Most of the studies that I have done have focused on different levels of biological organization. So the organization of this talk will start from the individual level and then go up to the community level. And so first, I'll talk about an example of an organismal response to ocean warming and acidification. And then I'll follow that up with an example of an assemblage response to ocean acidification, and then further zoom out and look at how we are working on quantifying the biodiversity and trophic connections along the Western Antarctic Peninsula, and then focus in on how we are using trophic biomarkers to trace some of these interactions on this geographical scale. So I'd like to start off by defining my terms, and for this talk, probably don't need to define climate change, but I'm going to be using it here in reference to the collective environmental impacts driven either directly or indirectly by the increasing concentrations of carbon dioxide in the atmosphere. Because these anthropogenic carbon dioxide concentrations have been increasing since the Industrial Revolution, when concentrations went from around 278 parts per million to over 400 parts per million today. And it's important to note that approximately 1/3 of this anthropogenic carbon dioxide has been absorbed by the ocean waters to date. And that will come up in a little bit. But first, people are probably most aware of carbon dioxide as a greenhouse gas and its influences on warming. And so I have this figure here to highlight some of the dynamics of warming across the globe. And on this figure, we have the change in temperature over a 10-year period from 1970 to 1979. These cooler colors represent a net cooling during this time period, and these warmer colors represent a net warming during the time period. And so I'm highlighting these because as we fast forward to the time period between 2000 and 2009, another decade chunk of time, you can see that we have gone from a predominantly mixed temperature change where you have both cooling and warming across the globe to a predominant pattern of warming. And most of the warming, as you probably are aware, is happening at the poles, particularly in the Antarctic and in specific locations in the Antarctic. And in the Antarctic, it is mostly centered, this increased warming is right along the Western Antarctic Peninsula. And that is where I have done all of my Antarctic research. I'm highlighting warming because polar organisms are generally thought to be very sensitive to warming. And so this figure here is just to highlight that. So on the y-axis, I have the aerobic performance and productivity of organisms on the right here. And then on the x-axis, I have the temperature range of organisms in different climate zones. And I want to highlight the polar organisms. Here they're termed polar stenotherms. So anything that is a stenotherm has been adapted to a very narrow range in temperatures. And you can see this area where they can do all of the activities, physiological activities required for life is pretty narrow, especially when you compare it to both temperate and tropical organisms. And just to give you a couple examples of what this looks like, here are two species of benthic organisms. I like benthic organisms a lot, so that's going to be the main focus here. I have an example of a brachiopod and a bivalve. And previous work has shown that both of these species are adapted to a very narrow range in temperatures. They can only survive in waters that are from -2 degrees Celsius to +3.5 degrees Celsius. It's a very narrow range when you're talking about these types of organisms. However, this is not necessarily true for every organism. There are several circumpolar organisms that have a bit wider range that they're adapted to, but there are a lot of species that have been studied have this narrow adaptive range. Now, I mentioned earlier that about 1/3 of the previously accumulated carbon dioxide in the atmosphere has been absorbed by the ocean waters. And that has been triggering what has been termed ocean acidification. And so ocean acidification can refer to the CO2 induced reduction in seawater pH. And so just in case you haven't had this your main part of your study, I'm going to go over the mechanism very briefly. And so basically what happens is carbon dioxide in the atmosphere combines with water. It will transiently form carbonic acid, which is a very weak acid. Carbonic acid, once formed, will quickly dissociate into a free bicarbonate ion and a free hydrogen ion. And it's the accumulation of these free hydrogen ions that causes an increase in acidity or a decrease in pH. Now you might be wondering, why has the pH not dropped dramatically in the ocean since we have been accumulating carbon dioxide in the atmosphere since the late 1800s? And that's because the ocean has a natural buffering capacity. The way this works is those free hydrogen ions are able to combine with free carbonate ions and thus make a new bicarbonate ion. So that negates that accumulation of those free hydrogen ions, so you get no change in pH. However, the ocean has been buffering this change for so long, and weathering of rock has not been keeping pace with the accumulation of carbon dioxide. So I'm mentioning the weathering of rock. That's a primary source for these free carbonate ions in the ocean is through weathering. And the pace of weathering has not kept up, so that now the balance is moving towards the accumulation of these free hydrogen ions. And so in Antarctic organisms, there has been some previous work on the responses of pelagic and benthic organisms. I want to highlight a little bit of that here. One of the most iconic Antarctic organisms is the Antarctic krill, Euphausia superba. And previous work has found that krill, when exposed to low pH or elevated CO2, they will increase their ingestion rate as their metabolic rate increases. And this is not, these effects are not just limited to adults. They found that there is reduced hatching rates when krill or eggs are exposed to the low pH that they are expected to experience as they sink through the water column. However, it's not all negative results for these krill. More recent studies have found that the lipid and fatty acid stores of krill are not sensitive to changes in pH that are predicted for the near future, which generally is considered to be around the year 2100 or even further in the future. So they've tested some pretty extreme pHs, and the lipid levels are not sensitive to these changes in krill. On the other hand, with benthic organisms, you may or may not know that there are lots of macroalgae that can be found along the Western Antarctic Peninsula, particularly towards the northern end. And there has been some work that has studied the responses of these algae to ocean warming and acidification. Just a quick note, I tend to go back and forth between using the terms macroalgae, algae, and seaweed. I mean the same types of organisms for each of those terms. Just a quick note, because I try to be consistent, but I know that I sometimes catch myself moving back and forth. So moving on, for this species here of Desmarestium menziesii, researchers have found that there were no changes in the photophysiology, fluorotannin production or growth in this species. For Desmarestium menziesii, fluorotannin production is their primary chemical defense. If you're familiar with temperate Desmarestia species, they produce a sulfuric acid compound as their defense, and that is not true for these Antarctic species. They rely on this fluorotannin production, and so they are the fleshy upright algae look like they are probably resistant to reduced pH and warming. However, the results for encrusting algae are a little bit more complicated, depending on whether you're looking at a calcified encrusting alga or a fleshy encrusting alga. There are some mixed results and interactive effects of both temperature and pH. So I've talked a little bit about the climate change. Let's talk about the animals that live here. So when you think about Antarctica, you're probably thinking of at least one of these organisms. These big, beautiful macrofauna, they are awesome and they're fun to see, but the ones that I get really excited about are the ones that live on the benthos. And so around Palmer Station, where I've done most of my work on the Western Antarctic Peninsula, approximately 80% of the benthos is dominated by macroalgae, particularly between the depths of 5 and 40 meters. So this macroalgal assemblage community is dominated by a mixture of red algae and brown algae. There are very few green algae that are found here. And many of these algae have accessory pigments that can help them adapt to the low light levels associated with living in a polar region. And in and amongst these algae, you'll find a lot of invertebrates. And so this is a picture of Desmarestia and some of the amphipod assemblages that associate with them. And so hopefully you've been able to find at least one amphipod in this picture, but if you haven't, I want to highlight the ones that I could find. So they are super abundant here. Previous work with this species in particular on the Antarctic Peninsula has found that if you clear a 1 meter square of the benthos and take all of the Desmarestia off, and you rinse every grazer off and meticulously count them all, there are over 300,000 individual amphipods that represent around 23 species. And so just to give you an idea of what that can look like, this is from one macroalgal collection where we rinsed off all of the associating amphipods. So they're a very, I like to say, vibrant and active community. And so I mentioned before that Desmarestia is chemically defended. So why are these amphipods hanging out on these macrophytes in such high abundance? So in this system, previous work has suggested that these macrophytes are producing chemical defenses that prevent predation. And so it's not only preventing predation from amphipods, it's preventing predation from other organisms. And so the thought is that amphipods are associating with these large seaweeds and getting a refuge from predation. In turn, the amphipods are providing epiphyte control. So even in the summertime, many of these, the vast majority of these big seaweeds are free of epiphytes. So they just don't have any epiphytic diatoms or filamentous algae growing on them. And so it's thought that these amphipods are the ones that are keeping that down. And so the macrophytes and the amphipods are getting a benefit from this association. And this has been termed a community-wide mutualism that is important for this region. All right, so I've been talking about the area between 5 and 40 meters. If we go deeper, then you find that the benthos is primarily dominated by these big invertebrate assemblages. And they can be very colorful and they're really amazing. So most of this work and most of the ecology that I've been discussing is based on work that was done here on the Western Antarctic Peninsula at Palmer Station, which is the smallest of the US Antarctic stations. At its max capacity, it can house 43 people. So it's a pretty small community. But one of the advantages of working here at Palmer Station is that a long-term ecological research group has been doing research here for over 40 years. And so there is a lot of data available for this region. And to set up my climate change experiments, I wanted to make sure that they were representative of the local conditions. And so I took some of the data recorded by the LTER and compiled it to get an idea of what conditions the organisms I wanted to study are being exposed to. So here, this is 5 years worth of data of the seawater temperature. So that this is the temperature of the seawater in degrees C. And you can see that there is some seasonal variation. It goes from around minus 1.5 up to almost 3 degrees Celsius in this 5-year time period. And that there is some day-to-day short-term fluctuations in temperature, but they're nowhere near the degree of fluctuation that you might see in a temperate zone. In addition to recording temperature data, the LTER also collects data on the chlorophyll A concentration to get a better idea of the phytoplankton communities in this region. And so I have chlorophyll A on the y-axis, and I just want to note that there is a break in this axis, and then time in months on the x-axis. And so you can see that the presence and abundance of the phytoplankton in the water column is pretty low for the majority of the year, especially during the austral winter time. But as soon as the sea ice breaks out, there is usually a very big bloom. And so you get lots of photosynthetic activity that then can remain elevated through the summer and then will go back down in the austral winter. So I took advantage of the fact that the LTER were collecting data points every week to get chlorophyll A, and I got them to also help me by taking bottle samples so that I could get pH and total alkalinity measurements to better get an idea of the whole water biogeochemistry. And so I found, so here I've plotted the pH on the total hydrogen scale on this axis, and you can see that the pH remains pretty constant, but then with that phytoplankton bloom, there is a big increase in the pH as the photosynthesis in the water kicks off and takes up that excess carbon dioxide. And then the pH remains high for most of the summer and then goes back down in the winter. And so I'm highlighting this and belaboring this point because I'm using these data as the basis for my treatments. So I used information from the Intergovernmental Panel on Climate Change, and I used their business as usual models to set my conditions. And I set them so they would represent predicted conditions that are supposed to occur by the year 2100. So to start off with the temperature treatments, I set 1.5 degrees C as my ambient temperature. And this is for 2 reasons. One, it's around the average temperature that these organisms experience in the summer. And it was also the temperature at the time that we were at the field station running these experiments. And then my elevated temperature predicted warming treatment is at 3.5 degrees C. This represents a 2-degree increase as predicted to occur by the year 2100. And then for the pH treatments, our ambient pH was set at 8.0. And again, that's because for the same reasons, or one of the same reasons as the temperature, that was the pH at the time that we were at the station. And it is close to that average or median pH that is experienced. And then our reduced pH or acidification treatment was set at 7.6, which again is predicted for the year 2100. And that gave us 4 temperature and pH treatments where we had an ambient, a decreased pH, a high temperature, and then a combined temperature and pH treatment. And so you might be wondering why we made life so complicated for ourselves to do these multiple stressor experiments. And it's because we want to get an idea of some of the interactive effects, because neither acidification or temperature warming are occurring in isolation. And so you can get information, you can have different combinations. And so you can have what's called an additive effect, and that's where the net effect that you observe is about equal to what you would expect if you added effect A to effect B. You can also see an increased effect, so an effect that's greater than if you just added effect A to effect B. And this is referred to as a synergistic effect. And then alternately, you can also have an effect where the net effect that you observe is less than if you add effect A to effect B. And this is called an antagonistic effect. So these, in this case, the two effects are sort of negating each other. All right, so that's why we did this. This is our reasoning. And for my PhD, I spent accumulated 12 months at Palmer Station, spread across 3 different field seasons. I quantified these responses listed here and more with these 4 target species, but for the sake of time, I'm just going to highlight a subset of these data. So I'm going to focus on these 2 amphipod species and 2 of the measures that I did with that. All right, so why did I work with these 2 species? First of all, Gondogenea antarctica, or what we in the lab like to refer to as gondo. It is a very common amphipod. It's small, it's very energetic, it moves around a lot, and it's an omnivorous amphipod. It's very abundant in these assemblages that I mentioned earlier. In contrast, Paradexamine fascicata, or Paradex, is a much larger amphipod. It tends to be more sedentary and is commonly found associating with just one macroalga. This one right here called called Plocamium cartilagineum. This is an interesting relationship because most amphipods tend to avoid Plocamium because it is strongly chemically defended. And previous work in the lab has found that paradexamine actually consumes the tissues of Plocamium and sequesters the chemical defenses produced by this alga. So these are 2 kinds of ends of the spectrum for amphipods, where I have a small, energetic omnivore, and then a more specified relationship with a much larger and more sedentary amphipod. And so to collect all of these organisms, we basically went out from Palmer Station in these small inflatable zodiacs, where we suited up and jumped in the water to collect everything by hand. And so I have a video here of what that looked like. This is a previous student, his name is Yusheng, and he's demonstrating how you do these collections. So we have these really fine mesh bags, and basically we gently dislodge them from the rocks and then capture them in the bag, being very careful not to dislodge anything that might be on the macroalga. So this is how we were able to target both paradexamine and Gondogenea. So once we got our alga and amphipod samples, we brought them back to the lab, where we acclimatized them for about a week before they were randomly placed into one microcosm that represented a combination of either temperature or pH treatment. So here you can see this is our experimental setup in the aquarium of Palmer Station. These are temperature baths that we used to control these water tables, and then each container had its pH controlled individually. And if you looked inside each of those containers, this is what it looked like. We had two, what we referred to as amphipod hotels. And we kept everything separate like this because we didn't want them to interact with each other and interfere with our other measurements. And so in one amphipod hotel, we had 10 paradexamine, and then the other one we had 16 Gondogenea. And we used these different numbers of amphipods because we were targeting having comparable amounts of biomass in each of those amphipod hotels. So that should tell you a little bit about the size differential between these two species. Simultaneously, we were also running an additional experiment looking at the responses of the fleshy macroalgae, and that's what these algae are here. And that is also another reason why we kept them in these hotels, so that they wouldn't eat our other experiment. So over the course of our 90-day exposure period, every day I would go in and I would check each hotel out of each of these 48 replicates. I would check each hotel and make sure that the amphipods were doing okay. If there were any molts, I would remove them. If any had died, I would remove them. And then I fed them every other day. And so now I'm going to show you some results of this experiment. All right, so on the y-axis, I have the proportion of amphipods surviving, and this is the time in days. And these are the results for Gondogenea. And each of these lines is color-coded for one of my treatments. And so you can see that the ambient and elevated temperature treatment had very similar survival rates. But as soon as you decreased the pH, there was a significant decrease in survivorship. So I mentioned the interactive effects that you can see from doing these multiple stressor experiments. And so here you can see that there does appear to be somewhat of a synergistic effect, where the difference between these two treatments is much larger than where we just raised the temperature. So there seems to be something going on when you add that added variable of decreased pH. So these are the results for Gondogenea. However, when we compare that to paradexamine, you can see that the trend is the same. There was a reduced survival when you reduce the pH, but it's much more dramatic. By the end of the experiment in this combined treatment, I only had 20% survival, which for me, it was really upsetting. I get really attached to my animals. But again, we have that same or similar trend where there's very little difference between that ambient treatment and the elevated temperature treatment, and then there's a much larger difference between the low pH and then the combined treatment. So there seems to be some type of synergistic effect. And so I mentioned that I also was collecting their molts at these same time periods. And so I have the molt results plotted here with the percentage of molts collected on the y-axis for Gondogenea, and then also for paradexamine. And you can see that there are similar trends where right here, there seems to be an increase in the molts collected. This happens to also coincide with one of our first big die-offs. And when we ran the statistics, we found that this was not by chance. And these, it appears that they're attempting to molt, and then they're not surviving the completion of the molt. However, those animals that did survive, with all of the other metrics that I looked at, there were no differences. So it appears that this survivability of this molt is pretty critical, but if they were able to survive, they looked similar in their biochemical composition and growth rates. And so one context we can put this in is by looking at and putting this in terms of resilience and resistance. And so just to define these terms, if you have some type of response on the y-axis and time on the x-axis, you have some type of disturbance, it will cause a decrease. So for instance, if these amphipods have this survival bottleneck, a lot of them die off, but then if they're able to rebound, then we would say that that population is considered resilient. However, if they don't change at all, that would be considered a resistant population. That means usually resistance can arise from several mechanisms, but one could be a response asynchrony, for instance. So that means that not all systems are crashing at the same time. And you can apply this context of resilience and resistance to the individual level or at the assemblage level. So I followed up my experiments with the individual amphipod species by looking at the amphipod assemblages. These included, well, grazer assemblages. So I looked at all of the crustacean grazers that I could find on the seaweed Desmarestia menziesii. So basically, we collected the seaweeds the same that I described, but this time instead of searching out individual species, we pooled all of the amphipods that were on those macroalgae into one of these fine mesh bags, and then we used a plankton splitter to split that volume and make sure that we aliquoted similar densities of amphipods into each one of these mesocosms pictured here that we were controlling individually. And so for this experiment, we tried 3 different pHs instead of just 2. We tried an ambient pH and then 2 reduced pHs, one that represents around 2100 predictions and then a farther future pH. And so our results look like this. So this is an NMDS plot. So points that are closer together are more similar to each other than points that are further apart. These black boxes represent all of the species that I identified found in the pH 8.0 treatment. So that's our control, and then 7.7 is the near future, and then 7.3 is our far future. So each of these dots represents the grazer assemblages, the species diversity that I could find or identified in each of the mesocosms at the end of a 30-day experiment. So I ran an ANOSEM to compare the similarity between these treatments, and I found that there were significant differences, even though those don't quite jump out in this NMDS plot. And it turns out that neither the 8.0 or the 7.7 treatments were different from each other, but they were both different from that extreme far future pH of 7.3. So let's look at some of the organisms that might be contributing to these changes. And so on this figure here, I have the mean number of individuals that I counted, and then on this axis, I have the organisms that I identified, which I also have many of their pictures here. And then each of these bars are color-coded based on the pH. And so I want to highlight some of these species. So these different colors are so these are representing those species that are most abundant that I found. And you can see that of the species that are most abundant, they all are not responding the same. So that you don't see the same trend in their abundance as you change the pH, which means they're having different responses. And that might be contributing to why we don't see a big difference between the 7.8 and the 8.0 treatment. However, when you color code this by feeding type, so brown represents omnivores, you can see that all of the omnivores are not responding exactly the same across all of these pH treatments. And then with here, I've got green, I'm highlighting the herbivores. The herbivores are not responding exactly the same, again, at all of these treatments. And so that is probably contributing to this resistance that we see to reduce pH, at least for that, which is predicted for the near future. However, there are a couple of caveats. This experiment was only run for 30 days. And if you're thinking that 30 days sounds familiar, it's because this 30-day period right here in my individual species experiments was right when I was having those peak molting periods. So it could be that with this shorter experiment, I was missing out on some of these changes in life history. And so in actually last year, in December 2019 and January 2020, we went back and we're set up experiments to do this experiment again with the same pH levels with 24 individually controlled mesocosms. Then we were going to have this run for 3 months. However, with COVID, that shortened our field season and it had to only go for 2 months. But we're still working on the results of these data and doing the necessary follow-up work because we haven't been able to get back to Palmer. And so we're following up on that previous result to see if we expose these assemblages over a longer time period, do we still see the same results? Okay, so now we're going to scale up from the assemblage level to the peninsula level. So I want to highlight again some data from the Palmer LTER. On this figure up here around Palmer Station, this little inset is representing the sea ice cover over a year. So on the x-axis, we have time in months, and the y-axis is representing different years. And then this color coding represents the sea ice cover at a given time. So you can see that over time, the sea ice presence and duration vary at Palmer Station, but there is a trend to that having that sea ice decrease. The other place where there is a lot of research done on the biology and ecology of the benthos, there's also has data collected for the LTER is at Adelaide Island at Rothera Station, which is run by the British Antarctic Survey. And here again, same color coding pattern. You can see that over time, the sea ice presence and duration varies, but there tends to be a decrease. And then down here in contrast at Charcot Island, this is the furthest south that the Palmer LTER hasn't been able to get to along the peninsula. And you can see, again, that same pattern of variation. We know that up here at Palmer Station, this is what the benthos looks like. It's dominated predominantly by these big brown seaweeds with the red macroalgal understories. At Adelaide Island, there is a lot less seaweed. There's a lot more fast ice present here at Rothera. And so it's thought that there is more sea ice scour and more shading. So there are seaweeds that are present, but they're not as abundant. However, we don't have really any idea of what the benthos looks like down by Charcot Island or this 500-mile region between Palmer Station and Rothera. So we do have some information, but the last time that somebody went and did a survey for this region was on the wooden vessel, the wooden research vessel, the Hero, in the 1970s. And so the data that they were able to collect was basically presence and absence. So you don't necessarily need to see or be able to read each of these little things, but each of these are macroalgal species and it's just presence or absence. And so in April and May of 2019, we had the opportunity to do a cruise to sort of fill in more of this data. So we're interested in getting a better idea of how sea ice cover and duration influences these benthic communities. And so I worked with a collaborative group that included a geographer who helped us use remote sensing to pick sites that had gradation in the sea ice cover, both for duration and for presence, for the amount of sea ice presence that is there. Each of these little red dots were potential sites that were identified before the cruise, and these green dots are the sites that we are actually able to get to. So at each of these sites, in order to document the biodiversity, we did horizontal transects at 5-meter depth, starting at 40 meters and moving up more shallowly. And then in addition to doing these transects, video transects to get an idea of cover and species diversity, we also did targeted collections where we collected macroalgae and invertebrate species for biomarker analysis. So we were using 2 different biomarkers. We are using stable isotopes and fatty acids. In addition to doing whole animal collections, we also did some suction sampling. So we basically vacuumed the ocean floor and got all the little bits that we couldn't grab by hand. All of these samples were brought back to the lab where collections were ID'd, dissected as necessary, cataloged, and preserved for biomass and diversity measurements. So using, taking genetic samples for genetic analysis and then also for trophic work. So again, this is another one of those studies that the results are still in progress. We're continuing to do the video analysis and genetic workups. However, preliminary results are indicating that the sea ice cover may not be the primary driver of the distribution of these different communities. And right now, by the end of this month, hopefully we'll be finalizing those fatty acid extractions. We'll have done around 1,000 fatty acid extractions across the sea ice gradient, and they include a wide variety of algal and invertebrate species. Okay, so I've mentioned biomarkers. I'm going to quickly do a brief overview of the biomarkers that I've been involved with working with. I've been working at the Oregon Institute of Marine Biology, where we've been using fatty acids as trophic biomarkers because there are a lot of fatty acids, which are the building blocks of lipids. Can identify anywhere from 30 to around 50 fatty acids in a sample. And they have different types. So you can have saturated fatty acids or SAFAs, monounsaturated fatty acids where you have just one double bond, or polyunsaturated fatty acids where you have 2 or more double bonds in the carbon chain. And so by identifying the fatty acids present, we can use them to the combination of the different fatty acids to trace nutritional sources through the food web. And so one thing to consider is that each organism will never look exactly like what it eats. And so for instance, this amphipod here eats this piece of algae. It has some trophic modification. It includes some kind of conversion or synthesis, and that will determine what parts of this diet are included in their body. And so it's good to directly quantify this for species. It reduces some of the error associated with making trophic connections. And so one of my ongoing projects is to quantify this for the amphipod species that we collected. And so for the experiment that I'm going to describe right now, I wanted to know whether Gondogenea, it looks like what it eats. Is it what it eats? So I fed them either a brown algae, a red, or this other chemically defended brown algae, or epiphytic diatoms. Each of these seaweeds and amphipods were all collected by hand, like I previously described. They were brought back to the lab where we sorted out the amphipods from everything else. And then we collected reference or initial samples for all of the diets. And then we freeze killed the Desmaresty inceps and the Hemanthophallus. That's because Gondogenea will not eat them alive, but they will eat them as they're decomposing tissues. And that's not because they're chemical defenses are breaking down. And so maintained these amphipods on monospecific diets for 9 weeks. And then after that time, we collected samples for fatty acids, which we brought back to Oregon, where we extracted the lipids, ran them through the GCMS, the gas chromatograph mass spectrometer, where we got plots like this, and I was able to identify and quantify the fatty acid profiles for the amphipods and their food sources. Okay, so here on this NMDS, this is like the other one where points that are closer together are more similar to each other than those further apart. But in contrast, on this one, I have overlaid a vector plot that is showing the fatty acids that are contributing to the distribution of these points, these multivariate points. And I would like to highlight a couple of these fatty acids as that are commonly used as diatoms for either, they're commonly used as biomarkers for either diatoms, red algae, or brown algae. And so here are those common biomarkers. So you can see that these biomarkers are showing up as driving some of this distributional shifts. So these triangles represent the algae, and these circles are representing the amphipods that were fed on the different diets. And you can see that the amphipods are grouping together. So it looks like they are taking up the signature of what they are consuming. And these wild amphipods are most similar to the brown algae, but they may be eating some combination of these other seaweeds, which is backs up previous work that has looked at stomach contents of these amphipods. And so we're continuing to work on this by performing additional feeding assays using additional macroalgal species and additional amphipod species to see if all amphipods are modifying the tissue that they're consuming in the same way. And this way we can generate reference libraries that we can use to better understand these trophic interactions along the Western Antarctic Peninsula across the sea ice gradient. Okay, so a quick recap. I've covered a lot of ground. We found a lot of mixed results for benthic organismal sensitivities to ocean warming. I presented the results for one of the negative effects that we found. We did find that seaweed-associated assemblages tend to exhibit a resistance to ocean acidification. We are continuing to work on evaluating the biodiversity and trophic connections along the Western Antarctic Peninsula, because presumably as the peninsula continues to warm, the southern end of the peninsula will start to look more like the northern end. And if we can understand how those changes are happening in the northern end, we can maybe predict what will be happening towards the south. And then we're doing ongoing work to figure out the trophic modification of these benthic organisms. So I'm going to jump into some acknowledgements. None of this work is ever done in isolation. I've relied on a lot of great assistance from in the field and at home universities. Most of the first work that I presented on the amphipods was done at the University of Alabama at Birmingham. The collaborative research along the Western Antarctic Peninsula was supported while I was working at the University of Oregon at the Oregon Institute of Marine Biology. And here are some of the amazing field teams that I have gotten to work with, and I would be happy to take any questions. Thank you.
Thank you so much, Julie. If anybody has any questions, please feel free to unmute yourself or type in the chat box.
Hi, Julie. Thank you for this great talk. I have a question about your experiment that you perform with the different pH and temperature for the different copper. And I was asking if your result has any relationship to hypoxia or oxygen level, and if it does also could be correlated to the abundance of algae in the areas that you sample.
That's a really good question. We did not measure the oxygen levels, but before the water went into each tank, we made sure that they were all oxygenated. So I don't believe that that would be limiting in this in our lab experiments, but it'd be certainly interesting to look at it in the field to get an idea if there is a buffering effect of associating with the macroalgae. That would be really interesting. That's not something I've had a chance to look at, but that would be a really good place to do some future research.
Very nice presentation. Thank you for that. I was curious that you were using EPA as a marker for red algae rather than diatoms. Most people would go straight to diatoms with that. I wondered why you chose that.
That's a good question. So it's mostly because EPA has been highly abundant in the red algae that I have looked at, more so than the diatoms. And you're right, that's one of the complications for using EPA as a red marker. A lot of the markers can be used in multiple contexts. Yeah.
Is there maybe a secondary fatty acid that you could add in there from the red alga that you could find? When we have trouble like that, we'll often go to sterols actually, because they can be quite species specific. They're kind of useless when you get up too many trophic levels, but at the bottom, they're actually quite species specific, and you'd be able to use your same extracts for sterols as well. Okay, that's good information to know. I haven't done that type of analysis, but it's something I'd like to learn more about. Yeah. Yeah, it's like fatty acids, only many fewer standards.
That sounds amazing. Yeah. Thank you.
I noticed that in your pH temperature experiments, which you showed first for your individual species, that actually the pH effect wasn't really very strong till about day 10 or 15. So do you think that has to do with the molding, or that they can take the low pH for a certain time and then after a certain time there's a problem?
I suspect that's probably right, that it's something that accumulates, that maybe they're able to physiologically just handle short-term exposures, but then as it gets more and more chronic, that builds up, and then over time, they're not doing as well. And I think that going through a molt process exacerbates the stressors, and I think that's where the bottleneck is. It's just like one too many things. Did that answer your question?
Yes, it does. Thank you. It's interesting so that all your molding happened at that time. So I don't know if these critters always mold basically at certain intervals, or was the low pH triggering the molting, or why did they all mold at that time when the effect was?
That's an amazing question. If you can help me get funding to figure that out. That's one of the problems with studying Antarctic organisms is very little is known about their life history and what is normal for them. And so in some ways, I'm kind of jumping into the deep end by trying to figure out what these impacts are without really knowing what normal looks like. And so that's one of the things I've struggled with some of this work.
Great talk. Thanks so much.
Thanks.
So, Julie, have you think about measuring any kind of metabolism, like biomarkers in those periods that you are showing there, like to see if there is any difference between their behavior, like in feeding?
With the feeding with the amphipods?
Yeah.
So I have done some of that work, mostly looking at their responses to the chemical defenses. And so temperature, well, I've only been able to really do that much with temperature. And I found that it will influence their feeding choices when they're exposed to acute temperature changes. But I did do some feeding assays with these amphipods at the end of the experiment using the seaweeds that were also exposed in this experiment. And I think my experimental design was a little bit too complex. It was hard to tease out any differences because I gave them 5 different feeding choices. But I would like to continue doing that. I think that's a really interesting area. So yeah, did that answer your question?
Yes, excellent. It's very, very interesting, and will be like, well, I'm not sure if you have any genes like for measuring expression of metabolism behavior, but because I assume that as a couples are probably a few, or not too much.
I don't have that information, but that would be really good. Sorry, I didn't mean to interrupt you.
Yeah, no worries. Yeah, that's very interesting. Thank you.
Okay, so these are the 2 species that I've done the feeding assays with the amphipods.
Okay.
So it's very limited, and most of that is time limiting. It's hard to get down there and do this, but it's definitely interesting to do.
Julie, I have a question.
Mhm.
In both of your experiments, did you ever decipher between male and females, or if any females were reproductive, or is that just too much?
I didn't, but that is something that ended up becoming a concern. So I didn't realize it at the time, but as I was pulling out the molts, I realized too late that a lot of them were females and that they had recently released eggs. They molt to release the eggs when they're reproductive. And so I think that some of this effect might be confounded by failed reproductive effort. And it looks like it was fairly, I hope it was fairly uniform that I had females across all the treatments, but I don't know. And I didn't have the ability to check for that because they weren't gravid at the time that I started the experiment. So that's a good question and something I would love to follow up on.
Is it easy to tell the difference, or would it have been too complicated to try to separate, the females, males, and females that did have eggs?
So you can do it, but it has to be done under a microscope, and these guys are pretty sensitive to handling. And so at the start of the experiment, I was trying to minimize my handling as much as possible because I didn't want any initial survivorship problems to be artifacts of my handling. So it's pretty easy once you stick them under the microscope. They have different morphologies. You can tell them apart. It just was a lot of handling. And when you think about the number of amphipods that I used, I had 48 replicates. In each replicate, I had 10 paradexamine and 16 gondogenea. That's a lot of amphipods to go through. And I was doing this all myself. This part of the experiment was just me.
Great. Thank you so much.
Yeah.
Is there anybody else? Okay, well, if there's no one else, thank you so much, Julie. We really appreciate you coming forward for the seminar. And everyone else, we will see you next week. Thank you so much for joining.
Bye.
Thank you, Julie. Thank you all. That was really fun. I hope that timing was good.
Thank you so much. Really enjoyed it. It was really, really cool to see, and to see some familiar faces. That was nice.
Yeah. All right. Thank you.
All right. Bye. Thanks.
Bye.